Okay, thank you, Sung-Ah and Dr. Park for the invitation. This is Christmas, so I'm gonna hopefully make this easy on everybody, make it easy to digest. So the talk is about a universal red blood cell production platform that we at the Bioprocessing Technology Institute at A*STAR has been working on for the last 6 years. To introduce BTI, the Bioprocessing Technology Institute, we are under the Agency for Science, Technology and Research and funded through the Ministry of Trade and Industry in Singapore, Enterprise Singapore, and the Economic Development Board in Singapore. Then we work very closely with our colleagues in the higher learning institutes, polytechnics, and some overseas institutes, as well as regulatory bodies like the Health Science Authority and the Singapore Food Agency to develop processes that will impact just gonna move all these things out of the way. Yeah, to develop a skilled workforce, technologies, and products and services for the biomanufacturing ecosystem in Singapore. So BTI is 30 years old now, and we work on several areas: product innovation, cell line development. Cell line development, in this case, we're going to talk about the red blood cell as the product. Through media development, downstream purification of separation technologies, ways to scale any process to larger volumes in order to mass manufacture, and then analytic science and technologies relevant to the processes. So a couple of years ago, the government invested 80 million in the boost for living cells as a medicine and projected the estimated market value of 5 billion by this year coming up. And I think we probably exceeded it already. But that shows how quickly this cell therapy industry is growing. And the goal is to deepen research in the digital space, the food tech, and the cell therapy R&D. So now specifically to the issue of blood shortages. Everybody knows that you get blood through donations. Typically 400 bags of blood are donated a day in Singapore, which is probably not much for a small nation compared to yours. But the thing is that the numbers of under 15 are starting to drop, right? It is in fact crossed over now to be less than the older folks, more than 65 years old. So you can see that the population that's going to be able to donate is not going to be able to meet those that need the blood. And in this graph, you see that as you get older, whenever you need blood transfusion, whether it's acute or chronic transfusions, you need more units when you're over 60 than if you're younger. And then, of course, the stocks of donations themselves went down during the COVID era, especially this rare blood type, AB minus, A plus, A at critical levels. So fortunately, we went into this space about, as I said, 6 years ago to try to solve this issue. And I'm going to walk you through 3 parts of the solution. One is making the starting cell line, which is the induced pluripotent stem cell starting material. Then developing a multi-stage differentiation process, and then finally doing some functional proof of the red blood cells. And there's one slide that shows some of the properties measuring using the Thoma cube, which I hope that will generate some questions. So the reprogramming method, this is on the top side, on the top level, is how you typically reprogram cells on a Petri dish. You reprogram cells, and after reprogramming, you see emerging colonies, and then you pick them into individual plates, wells, and then expand them up. There's a lot of manual handling, right? And then you bank them. This is the platform that we have developed, where we take cells that are somatic cells, then we reprogram, but we selected onto these microcarriers. Right? So they would only pluripotent stem cells would bind onto the microcarriers, and then we spread out these microcarriers. We pick individual microcarriers into a 96-well plate. On this plate, we get many more candidates than in this because we're picking individual microcarriers. And then the ones that are positive, we add more microcarriers, and then they become larger and larger clusters. So we can bank them, and we can also direct them towards the lineage of choice. Because when you make the pluripotent stem cells, you want to be able to check that the cells can become the target cell that you want, not just be a starting pluripotent stem cell state. So our method shows that we can reprogram about 30 times more candidates than the traditional monolayer method. Efficiency here as well as the total number of positive clones. So these shows 12 such clones that we picked, shown by these three markers. We call it a surface marker, TRA160, an antibody 84, and then an intracellular transcription factor called OCT4. So they're expressing high levels of these, over 90%. And then we pick 2 of these and differentiate them to 3 germ layers. So these are like the muscle type, the neural type, and then the endoderm, which is the endocrine organs of the body. It shows that they can form the 3 germ layers. You also need to check that genetically that they have not been abnormal. So these are checking the 23 pairs of chromosomes. You can see they're normal. So you got the 2 pairs of chromosome 1 all the way to 22. And then you have the X and the Y for the male, and then the XX for the female. So these show that these 8 candidates that we picked are normal pluripotent stem cells. Importantly, because this technology allows to in parallel differentiate them to the target lineage, we chose to make them into heart cells, cardiomyocytes, and the blood cells, which is the erythroid lineage. So again, we see of the 12 candidates, most of them can form these heart cells at a high level expression of a cardiac marker, CTNT. And erythroid differentiation, you can see they can also form erythroid. And then 2 of them are extremely good to expressing 80% of this marker, BRCA5. So if we were to pick clones to move forward for therapy, for example, we would look at the ones that are capable of differentiation to the heart lineage and the erythroid lineage. So you see candidate 3 and 7 are capable of doing both these types of differentiation. So that's the advantage of having multiple lineage differentiation post making the reprogrammed cells. Now we move on to the manufacturing process. And so this slide essentially summarizes all of the capability of the group that has developed. We can reprogram cells from 4 different sources. So these are different types of somatic cells, T cells, skin cells, blood cells, and mesenchymal stem cells to these iPS cells. Once we've got these iPS cells, we then can expand them in culture. And here's expansion reaching 3 million cells per mil, which is a high titer. And then in different periods of time, we've made them into neural stem cells, eye pigment cells, heart cells, beating cardiomyocytes, blood stem cells. And today I'll elaborate more about these red blood cells, erythroblasts and enucleated red blood cells. So you can see these are the beating clusters of cardiomyocytes in culture. So erythroid differentiation is probably one of the most complex and highest density cultures in the human system, in the developmental space. Right? You need to make a lot of it to circulate around the body. Plus, you need to take it through multiple stages of differentiation. So this is a schematic to show you essentially what we've done. We take these primary cells, we make them into iPS cells. And the marker that we have to pick up is the 4 markers of 4, SSCA4 and FAR116. Then we induce them into a stage called mesoderm and the blood lineage or hematopoietic commitment. And they have to express these 3 markers, CD34, 43, and 45. Then from this stage, we have to direct them further, because from here you can make B cells, T cells, platelets, and so forth. We have to specifically direct them down to the blood lineage. So these are different stages of the erythroid lineage. And one of the prime markers is CD235A. And then they become reticulocytes. And then when they eject out the nuclei, they become these normocytes or nucleated red blood cells. So when we started with Jay, who's really been the driver of all of this work, we tried methods using clusters of cells, just the clusters of balls of cells. We call them embryonic body, and tried to make mesoderm. But these clusters really couldn't hold together. Once they were induced to form these early stage hematopoietic precursors, they stopped here and then didn't mature out. So in came our method when we created these clusters of cells on the microcarriers and then induced them to mesoderm. Fortunately, they held together. There was minimal cell loss and actually growth in the EB embryonic body structure. And when these were replated, they did form hematopoietic stem cells. And then these hematopoietic stem cells could further differentiate to erythroblasts, and then finally enucleated red blood cells. So that was the first breakthrough, 80-fold improvement in production of erythroblasts versus a very poor method. Then the next paper showed that we could do it for multiple cell sources. So this is the human embryonic stem cell line, and then these are different donors of induced pluripotent stem cells. So we show we could select for and grow up these induced pluripotent stem cells, and then they're normal karyotype, they have high expression of pluripotency and so forth. And then here are 6 examples where we showed that it could become the erythroid lineage. And importantly, of these different donors, one of them expanded very significantly to 60,000-fold towards the erythroid lineage over a period of 42 days. So this is the clone then we took forward for process development. So while we could do it in the 6-well plates, we needed to demonstrate we could do it at least in a simulation of a bioreactor. So in the same pot, essentially, we're growing up the starting cells for 7 days. Then we induce it by changing to the media mesoderm lineage for 3 days, and then another 14 to 21 days in this erythroid lineage media, and finally maturation. So we showed we could do all of this. So here's the expansion, again, 3 million cells per mil. These are the pluripotent markers of these expanded cells at the end. And then during the 3 days of induction, we could see these hematopoietic genetic markers when we, also in the mesoderm, we showed this expression of early mesoderm markers. And then as we move towards the next stage of erythroid hematopoietic differentiation, you see these CD34 positive markers go up. These are the clear bars, and then they drop down. Then the CD43 and 45 dark bars start to go up. which is moving towards the erythroid limit. So we see also these are other genetic markers. And then post differentiation towards 28 days, we see the CD235 markers go up towards 80%. So we're getting these erythrocytes. And in terms of cell numbers, we ran multiple of these suspension cultures. And here we're hitting about almost 20 million cells per mil, about 17 million cells per mil with the X13 clone. However, even at these high densities, 10 to the 7 cells per mil, to make one bag of blood, which has 10 to the 12 total cells, you need 100 liters of volume. So our goal was to push this yield to 10 times that, which is 10 million cells per mil, meaning that we would then need a 10-liter bioreactor to make a unit of blood. So we're still very far from what the body can do, but that's our goal right now, to push it towards this 10 to 8 cells per mil. So we also checked for the expression of hemoglobin. So you can see that the spinner cultures are producing the adult beta hemoglobin, alpha hemoglobin, and then also expression of this gamma fetal hemoglobin, which you can see also in cord blood, but you don't see in the adult blood. Importantly, we also show that the cells can carry oxygen. So there's an oxygen binding curve, which is closer to the cord blood behavior, the P50 release values compared to the adult red blood cells, which is a value of 17.9. The last stage of these erythroblasts is they must enucleate to actually function as the oxygen carrying entity. So when we were working on this a year or so back, we had to combine it with feeders. We call these OP9 feeders. So you can see that these are the cells which were cultured out. Sorry, just not seeing my screen. Yeah, here we go. So this we took erythroblasts at different days, right, of that growth, at 25-day-old cultures, 28-day-old cultures, and 35-day-old cultures, and matured them up with feeders. So we can see that over 7 days, maturation. Yeah, over 35 days of maturation, we get 65% enucleation. And same with 28-day-old erythroblasts, we're also getting 62% enucleation. So these are the ejected nuclei from out of the culture. However, if you took erythroblasts that were too old, say they were already 35-day-old in culture, they did not efficiently enucleate, and they ended up giving only 33% enucleation. And here we show that when we tried to enucleate without OP9, there were more more dead cells. So you can see all these are dead cells, and very few clear red blood cells. Whereas in this case, there was a high percentage of non-apoptotic cells in this, which is annexin negative, and there were more enucleated cells. Similarly, here we show that we get higher enucleation with the 2D. If we try to enucleate without this feeder cells, we were getting very low enucleation, 5.9% versus 41 to 62%. And importantly, when we combine the feeders with mixing, we got more enucleation of these red blood cells. So, and then we looked at some of the biophysical measurements. To finally make the product, we actually need to get rid of all the nucleated cells and leave only the enucleated sample. So a simple non-woven filter would remove most of the nucleated cells and give you the final product. And these are the SEMs. I'm told by Benoit, who is my collaborator in this space, that these are normal looking cells, and the crinkling does happen from time to time. Okay, so here's the tomocube analysis, which I guess I need expert to comment on. This is work done by Benoit, my collaborator. And you can see that, interestingly, when we compare, this is just regular adult blood. This is hematopoietic stem cells that were differentiated into blood. Then these are cord blood red blood cells. And then these are our iPS cells. So the cellular volume seems to be smaller for these donor-derived blood, whether you get it from adult or cord blood, and then even differentiation from CD34 cells. Whereas our iPS-derived samples, 2 of them, had a larger cellular volume. In terms of the surface areas, it seems also about 50% more surface area here between these 2 samples compared to the adult and the cord blood. Hemoglobin level gram per deciliter is in the around 20-25 range, which I assume this is normal. Sphericity is again around the same. Hemoglobin content picogram. So we got larger cellular volume, and we also have a larger content of hemoglobin. And then the membrane fluctuation is marginally higher for these iPS cells. So my take on this is that potentially we have a better quality source of cells because we're making them fresh from essentially an induced pluripotent stem cell state. So you get larger cell volume, larger surface area, a higher hemoglobin content. And we haven't even looked at things like the mRNA, the microRNA, and other stuff that we know red blood cells carry as well. And these contents may actually have vitality, youthfulness properties, which we would like to test once we can generate enough of the material. So going into the genetic analysis, this is a volcano plot to compare between the normal blood and our iPS-derived blood. So the key take home here is there's a lot of still apoptotic genes expressed in the iPS-derived cells, higher signaling. Cell cycle genes are also very high. These are all the red ones. Cytoskeletal genes also heavily red. Expression of hemoglobin is high, and then mitochondria is high. So these 2 are also indicative that if in the erythroblast state, there's a lot of mitochondria. It could be that these are also good organelles that may be useful for therapy of some sort. So finally, this is the paper with the details published in Stem Cell Reports 2021. Our hope is to get into these bioreactor 50 liters. This is Benoit, who did most of the characterization and is also developing the mouse animal models, and Alan, who's supporting a senior scientist who's supporting the group. So our goal is to move from this, donating blood. You get different age of blood from different donors. Moving forward, you probably get older donor blood, and to be able to essentially mass produce in a bioreactor the required unit of blood. And if the quality of the blood is better, right, it may be we don't need a full unit of blood. We may need only half of it or 1/10 of it because the cell quality is better. So with that, that's the summary of the work. We developed this technology that can isolate more iPS clones that can proliferate and differentiate to blood. And then all of this work was done with universal red blood cell donors, scaled to bioreactor. And these cells are functional and maybe of better quality than donor blood. All right, so thank you. That's my contact if there's any questions.